I'd like to thank CureOM and Sarah specifically for the invitation to come share with you guys today. Today I will talk to you guys about adjuvant therapy and clinical trials, kind of in broad strokes. So when we talk about adjuvant therapy, the word adjuvant actually has two definitions. The first definition is the one we're talking about here. It's treatment given to prevent recurrence or the spread of cancer. It's known as secondary prevention. Primary prevention is how to prevent the cancer in the first place. And with uveal melanoma, we don't really have causative factors that cause this melanoma, so it's really hard to implement primary prevention. But once you have the disease, we try to aim strategies at preventing recurrence or metastasis. So that's secondary prevention. But adjuvants also exist in the vaccine world. And so these are agents used to modify vaccines to either make them more stable, to make them more powerful at delivering their target molecule. And so when you think of the measles, mumps, rubella vaccine, the adjuvant in there is aluminum-based. And so that's, been in the news the last couple of decades. And in cancer vaccines, we often use water and oil emulsions, though there are some new other kind of adjuvants that can be used, toll-like receptors, et cetera. The most common ones in cancer vaccines are things called Montanide or Freund's, incomplete Freund's adjuvant. So we're talking about adjuvant therapy, treatment to prevent recurrence. So we have to usually think about this on a trial, a clinical trial, but how do we design these clinical trials? So, you can often just think of a single-arm, open-label trial where we take essentially the high-risk population, however that's defined, you give them an intervention, and then you look, in this disease, we often look at relapse-free survival at 3 years because that's kind of a landmark time point, or you want to look at overall survival. I'll tell you the FDA, the agency often requires an improvement in overall survival, not necessarily just in relapse-free survival, in order to consider adjuvant therapies for approval. So depending on how much improvement over, say, a historical control, a trial like this would take anywhere from maybe a few dozen patients, 33 patients, to upwards over 100 patients. So more favorable to the agency is things that are randomized. So where you take a high-risk population and you randomize them to either intervention or the gold standard, and right now there is no gold standard in uveal melanoma, so that would be considered observation. So a study like this increases the number of patients you need to enroll upwards to 500 patients. And in talking to groups like this over the years, we know there's not a lot of enthusiasm for knowing that you have high-risk disease and being randomized to observation. And so that conversation has been had over and over with groups like the FDA, yet they still really say, how do we know this is any better? We don't just want you to compare it to a historical population from 10 or 20 years ago. So that's what we struggle with. So instead of 1-to-1, we've often thought about 2-to-1 randomization. So 2-to-1 means there's 2-to-1 chance you get randomized to the intervention rather than the observation. So instead of a 50-50% chance, it's more like 67-36% chance. But still, you still need a really large number of patients to enroll a study like this. And again, if the observation group eventually drops out or discontinues from the study follow-up period, it becomes inevaluable at the end because you have this fully enrolled intervention arm and an incomplete observation arm to compare. So, and then you think about things like multi-arm randomization. Why are we just comparing one intervention? What if we just compare, these are just 4 examples. These are not necessarily the 4 best agents. But if we're comparing, a c-MET inhibitor versus a MEK inhibitor, an HDAC inhibitor, immune therapy, if you're actually comparing between all these things that may be equally efficacious or so, the enrollment skyrockets, actually well past 1,000. But if you just say each arm is individually, it's a non-comparison study, you still require several hundred patients to do a study like this. And so there's no perfect model in a rare cancer. Typically, most of the studies that we think of in the adjuvant setting are done single arm, open label. And part of what makes a good adjuvant trial is figuring out the right population. You don't want to treat every uveal melanoma patient because there are low-risk patients, and those patients may actually not need an intervention. You want to figure out what you're targeting. Are you targeting a gene that turns on the cancer? Are you targeting a faulty immune system? What is it you're targeting? And then, as we said, you need to design the right trial. So we take a lot of what we know about melanoma from skin melanoma, and adjuvant therapy started with interferon. It moved to pegylated interferon. And actually, we studied this in mice for some time in uveal melanoma, and then moved it into humans. So this was a study from some time ago that looked at about 121 patients in one intervention group compared to historical control. They received 2 years of adjuvant interferon, and these were self-injections that the patients would do 3 times a week. They followed this group of patients, it was about over 100 patients, for almost 10 years. And they found when they compared it to, again, historical control, there was no difference in how often the melanoma actually came back, but more importantly, if they lived any longer. And so interferon really was thought not to be terribly effective on its own. And so the group at Cleveland Clinic said, well, there's some evidence if we add chemotherapy to a round of, or a course of interferon, we may actually get better effect on circulating endothelial cells and melanoma cells. And so they ran a study several years ago looking at essentially the high-risk monosomy 3 population, and patients had to be enrolled within 8 weeks of their primary therapy. And they received a couple of doses of chemotherapy followed by interferon, this interferon alpha, and they received that for 6 months beginning 2 months into therapy. This study has been completed, and we're just waiting results to hear how it did. And then most recently in the skin melanoma setting, there's adjuvant ipilimumab, adjuvant nivolumab, and as of last week, adjuvant pembrolizumab data showing effectiveness in skin melanoma. And so again, we take what we can from the skin melanoma population and learn. And we ran a phase 1-2 study of ipilimumab for the high-risk uveal melanoma population. This was supposed to take high-risk patients defined as the class 2 population, or the population who had monosomy 3 with a thickness in their tumor greater than 8 millimeters. Patients were, it was a dose finding, and so the first 3 to 6 patients received ipilimumab at the standard dose of 3 milligrams per kilogram, and we saw no toxicities, and so the subsequent patients were enrolled at 10 milligrams per kilogram. It was a year of therapy, and so after the 4 doses every 3 weeks, which is the induction period, they received maintenance period up to a year, so essentially 3 maintenance doses. And our original design for this study, the class 2 cohort and the monosomy 3 apical, looked to enroll about 90 patients, about 30 some in the class 2 and about 50 in the monosomy. So we were able to enroll 10 patients, and then as happens in the modern world, our funding partner decided to go a different way with the funding, and so canceled the contract, and we had to halt our treatment at 10 patients. So all of these patients enrolled in the year 2013, and so now we've had our median 3 years of follow-up. And as you see, the first 3 patients went on at 3 milligrams. They had no unexpected side effects, and so we were able to put the rest on at 10 milligrams. As you look at this column that says grade 3-4 adverse events, you don't see anything that we didn't know from the skin melanoma population. So we know things like diarrhea happens. We know that elevation of your liver functions happens, rash, itching, and then things like a vasculitis. And 2 patients did end up not finishing the year due to these side effects, and the remainder of patients finished. So we're hoping to take a look at what the recurrence in this is, but this is really going to be a difficult study to publish. It was originally planned for 30 patients in this class 2 population and 89 overall, and only enrolling 10 patients will be statistically criticized. Why are you just publishing on 10 patients? And it's because the funding, the drug manufacturer stopped providing the drug and the money, et cetera. So what can you do in that situation? So there was also a hepatic irradiation study many of you guys might have heard about. I think it was run out of UCLA, where the high-risk patients would undergo weekly radiation, or daily radiation, I should say, over 2 weeks. So it was 10 fractions to the liver. It was a preventative sort of dose. So if there was any micrometastatic disease sitting in the liver, you would get radiation to that. They had planned to enroll 50 patients, and their study was terminated after 1 year for slow enrollment. They were able to enroll 2 patients, and unfortunately, what happens at each of our hospital institutional review boards and even the FDA, if you have a slow enrolling study and you don't really have justification for why it's enrolling so slowly, they have the right to shut you down because you're using institutional resources, et cetera, to keep a study open. There was a peptide vaccine study that was a multi-peptide targeting at some of these 4 proteins in melanoma, so MART1, GP100, tyrosinase. These are all proteins that are expressed in skin and uveal melanomas. And this was a really large phase 3 study. Patients were going to be randomized to either receive the vaccine schedule or a year of observation. And 600 patients, it was originated out of Denmark, and this study was also terminated due to slow enrollment. So where are we now? What do we have open now? There's a couple of trials in the US that are open. One is the crizotinib study that's run out of the Northeast. That is also struggling a little bit with slow enrollment. This is for class 2 tumors who have their longest tumor dimension is 12 millimeters. So that can be either your basal dimension or your apical height. I think that one of the challenges is you have to enroll within 90 days of having your primary treatment. And so the primary patients, you guys can attest in that first 3 months of having your primary treatment, you're going still to the eye doctor for your follow-ups. It may not be a great time for you to be traveling or starting these adjuvant therapies. And so part of their difficulty enrolling may have been that tight window. It is almost a year of treatment. It's 48 weeks of treatment or 12 one-month cycles of an oral drug called crizotinib. And then there's another study out of Thomas Jefferson, that's the sunitinib or valproic acid. Patients will get randomized to either a year of 6 months of sunitinib or 6 months of valproic acid. And these are for patients who have monosomy 3 with 8q amplification or for the class 2 tumors. And you have to start this within 6 months of your primary treatment, so a little more liberal. And the 2 arms are not being directly compared to each other. So it's not asking, is one of these drugs better than the other? It's just looking to establish at 2 years how many patients have progressed, and at 3 years how many patients have progressed. So, but you'll never be able to finish the study and say that, one drug is superior to the other. It's really just information gathering for the 2 arms. So where are we going moving forward? We are not stopping designing adjuvant trials. It doesn't matter that the funding is difficult. It doesn't matter that enrollment is slow. That tells us we just haven't designed the right trial yet. And so we're still looking into adjuvant peptide vaccines. The ones that the Copenhagen group did, the 4 peptide, that was a nice package. It's probably the trial design that was difficult. There are other protein or peptide targets that make sense in this disease. And so just like there's always a new flu vaccine every year, we'll never really be done creating cancer peptide vaccines. And so that may be something to consider to boost the immune response against microscopic circulating disease. We are designing with Georgetown, Columbia, and some other hospitals a year of adjuvant nivolumab plus ipilimumab. And so this is a treatment that's approved for skin melanoma in the metastatic setting. And as many of you know, we've also been running a trial in uveal melanoma in the metastatic setting. So now the question is, can you take this package of drugs for a year for prevention of disease? And then many of you are familiar with the company Immunocore, and they have a compound known as IMC-GP100. And so this drug is making its mark in the metastatic setting, but there may be room to move a drug like this into something like the adjuvant setting. And as many of you guys may know, that these types of things, and sometimes even peptide vaccination schedules, they do restrict you to having a very specific immune type, and that's an HLA allele. And that's because these vaccines or peptides or molecules are recognized in the context of that immune system. If you don't have that circulating immune system, then you don't process and present this drug or vaccine properly, or the way that the immune system knows to receive it. And so it will just essentially be eliminated from your body. So there's also laboratory-based trials, and what we mean by that is, you might not necessarily be getting an intervention, or you may be getting an intervention that's very difficult to find out if it's working. And one of the classics here is the HDAC inhibitors, so valproic acid and vorinostat. One of the difficulties is we don't even know the right dose to use, period, for this disease. Those drugs are approved for either, neurology indications or skin T-cell cancer indications, and it's not clear what they're doing in those diseases is the same as what we need them to do in uveal melanoma. In uveal melanoma, we use HDAC inhibitors to essentially express the melanoma. The melanoma exists in a sort of a seed-like form, and you don't know what flower that's going to bloom into in the body, and so these HDAC inhibitors are given to sort of push it into blooming. So in that regard, that's not the same indication or how they're used in those other diseases. So this sort of, T-cell lymphoma dose of vorinostat may or may not be the right dose for uveal melanoma acetylation, which is essentially what you're doing. So when you're in the adjuvant setting, there's nothing to measure or follow, right? There's no scan to follow to say something's getting bigger or smaller. We're just timing you to see if and when the cancer comes back. So that's just a wait and see game. But there are assays we can do or blood-based research we can do to say at this dose of HDAC inhibitor, are your peripheral blood white cells, undergoing acetylation? And so that would be considered a lab-based trial, not really sort of an interventional treatment trial. And then there's this buzzword in cancer right now, liquid biopsy. So liquid biopsy means, can we take a blood sample and see if there's cancer in the blood sample? And there's several different ways to do this. You can look for whole tumor cells, such as uveal melanoma whole tumor cells, or you can look for the things like the GNAQ mutation, the GNA11 mutation, and so you're looking for free-floating DNA in that regard. Again, that's not really an intervention. You're not getting a treatment, but you would be followed to see if there's different changes in these levels over time. And then microbiome, another buzzword that's been all over Forbes and Science in the last year. Microbiome is the bacteria in your body. And you have microbiome, mainly in cancer, we study it in your mouth and in your gut. And so we're studying the microbiome to see the bacteria in your gut really has a role with your immunity. And so there are patients who do very well on immune therapy that we're looking to characterize their microbiome. And then the patients who maybe aren't doing so well with immune therapy, are they lacking certain bacterial strains that we see in the good responders? And essentially, probiotics that everybody likes to take and buy off the shelf, these probiotics change the concentration of different bacteria in your gut. So right now we don't know, what's the winning combination of bacteria to take, and so it's a little hard for us to just recommend a probiotic. They're all different, they all have different strains, and your content may be different, than the probiotic. The probiotic may actually change the content of your microbiome. So we're studying this, and it's not uncommon for you to come to MD Anderson, and we'll talk to you about collecting your stool or swabbing your mouth, and again, not an interventional trial, but a laboratory-based trial to see if there are changes. And then my last slide is really just to talk about what to do when you're offered something on a clinical trial versus off protocol. Off protocol is often what we say when something's not done on a clinical trial. So there's pros and cons to both, as you guys can imagine. If you look at clinical trials, the pros are that most of the time this is covered by insurance. It's very rare for me, I'd say since 2000, probably 14, it's very rare for me to get an insurance company saying this person doesn't have coverage for clinical trials. I remember in 2011, I had this one police officer with skin melanoma, and his, Fraternal Order of Police insurance didn't cover clinical trials for cancer. And at that time in skin melanoma, the best treatments were on clinical trials. And so it was really, really difficult trying to get by with just chemotherapy on him. So most clinical trials will be covered by insurance, but more importantly, the study drugs are often provided for free. That is actually the bulk of a clinical trial cost to insurance. And so if they're picked up by the sponsor, and this is not an absolute, there are some studies that do require the insurance to pay for the drugs, but if the study drugs are provided, that's often a benefit. They're often incredibly science-driven. This idea that you're a guinea pig on a clinical trial just really isn't a valid statement once you're in a cancer trial, specifically if it's for uveal melanoma. And even if it's in phase 1, you're not really truly a guinea pig. There is a lot of scientific rationale about why that molecule medicine or treatment schedule was chosen. When you're on a clinical trial at a hospital, you often have a research nurse as your point person. So, you all have clinic nurses, but that clinic nurse often also has 150 or 200 other patients. When you're on a clinical trial, you're often one of 10 patients that research nurse is following on that particular clinical trial. So you have more of a direct advocate to report your side effects, et cetera. And your side effects are really carefully captured on the clinical trial, and they're compared to the other enrollers to see if there's any signal about side effects. The sort of downside of clinical trials is they often have a lot of visits in there, and as many of you know, there's often a lot of blood collections and things like that to see is the drug getting into the blood, et cetera. There's a lot of compliance rules, and so it's rarely happened, but if a patient is really non-compliant with taking the drug, keeping the visits, seeing their local doctor, getting blood work, the clinical trial does have the ability to sort of terminate their participation for compliance reasons. There's also really strict eligibility criteria, and sometimes they seem kind of arbitrary. You know, why on certain, we used to actually on our immune therapy trials exclude patients. There used to be language that said you were excluded if you had autoimmune disease, and there was no further language explained than that. Well, type 1 diabetes is a type of autoimmune disease, but that's not typically the exclusion that most sponsors had in mind. And so the clinical trial eligibility can often be strict and a little bit inflexible unless you have a sponsor that's willing to sort of make exceptions. And then there's also the possibility of discontinuation of the study or drug. As I explained to you earlier, you know, we had no control over a study being discontinued by a sponsor, and it was a study for another year. We kept getting patients coming saying, we're here for this study, we're here for this study. And it's because, as you guys know, the clinicaltrials.gov website is a federal website, and it's very slow updating that website. So you have no control over whether something is sort of going to continue to go or be offered when it's under a clinical trial. You have little control, I should say. And doing things off protocol also has its pros and cons. You know, the pros are often, you can make the exceptions. If a trial said, hey, we don't want to take anybody with rheumatoid arthritis, you could accept them if you were doing something off protocol, understanding why they were excluded in the first place, would side effects get worse, et cetera. You can often be more flexible in the schedule and say, hey, listen, we're going to work around Hanukkah this year, or Christmas, or graduation, et cetera, where a clinical trial is not always that flexible. But the problem is these are not always supported by insurance. And so you can say, listen, I think there's a lot of rationale to give somebody an HDAC inhibitor for uveal melanoma, and the insurance is like, great, they can pay out of pocket for it. You know, so that's an unrealistic expectation, and these drugs are really costly. As you guys know, this is now a weekly sort of news blurb about how costly cancer drugs are. The data outputs, how do you even interpret, you know, 4 patients who received a treatment not on a clinical trial? There's a lot of sort of, I'd say, criticism that happens when the study team tries to publish that. They'll say, well, you cherry-picked the 4 best patients or the 4 healthiest patients, or, you know, et cetera, et cetera. And so if the data that comes out of an off-protocol use of drugs is not always validated. And the side effects are not often captured on clinical trials that report to the FDA. You have to present these annual reviews and review all the toxicities that each patient has had. On an off-protocol treatment, you never have to do that. And so it's just you're only reporting what you've, you know, sort of thought to capture. And you don't really have a point person when you're doing something off protocol. So you'll call that study, that clinic nurse, but you're, you know, patient number 150 to her. They care about each and every one of you guys, but they're not as balanced about taking care of patients that are off protocol. So that's kind of all I had as far as adjuvant therapy. I do want to acknowledge all the patients and families and caregivers who participate in the clinical trials in the adjuvant and even metastatic settings. We know that's not an easy ask, and we do appreciate it. And I also just want to acknowledge all the people in the room who we collaborate with inside of MD Anderson, outside of MD Anderson, and all my colleagues in the uveal melanoma field.